Welcome to session 36. The topic of today's discussion is enzyme kinetics. What is enzyme kinetics? So, kinetics is the study of the rate at which compounds react. So, the rate of enzymatic reaction is affected by enzyme concentration, substrate concentration, effector molecules, and temperature, and even pH. Michaelis and so this is the portrait of Michaelis Menten, Leonard Michaelis and Maud Menten. So these two people, they have derived an equation called as equation. We'll look into that equation. Why to study enzyme kinetics? Quantitative description of biocatalysis to determine the order of binding of substrates, the rate at which the substrate binds, to elucidate acid-base catalysis, to understand the catalytic mechanism, to understand the rate of reaction, uh, to find effective inhibitors, and to understand the regulation of the activity, to understand the regulation of activity. And even this kinetic studies are very important in preclinical research and in pharmacology. So Michael maintained they proposed a hypothesis. Michaelis and Maintain, while studying the hydrolysis of sucrose catalyzed by the enzyme invertase, proposed this theory. Their theory is, however, based on the following assumptions. Only a single substrate and a single product are involved. The process proceeds essentially to completion. The concentration of the substrate is much greater than that of the enzyme in the system. An intermediate enzyme substrate complex is formed. The rate of decomposition or the breakdown of the substrate is proportional to the concentration of the enzyme substrate complex. So these are the assumptions of Michael-Menten equation. So the derivation of the enzyme kinetic equations, it starts with a model mechanism, identify constraint and assumptions, and carry out algebra. The simplest model mechanism is we have a enzyme plus substrate, and it forms an enzyme substrate complex, and this gets converted into enzyme plus product, and it is one reactant one product without inhibitors. The theory postulates that enzyme forms a weak bonded complex, enzyme substrate complex, with the substrate. The enzyme substrate complex on hydrolysis decomposes to yield the reaction product and the free enzyme. So this is the reaction. Uh, enzyme plus substrate gives rise to enzyme substrate complex, and we get free enzyme plus product. And this is the famous equation called as equation. Velocity of the enzyme substrate catalyzed reaction, V is equal to V max into substrate concentration divided by Km plus S. Uh, so where V is equal to velocity or speed of the reaction, and V max is equal to maximum velocity of the reaction, right? It is the maximum velocity of the reaction. S is the concentration of the substrate, and this is V, and Km is the Michael-Menten's constant. Okay, Michael-Menten's constant. Uh, this particular equation can be used to calculate Km after experimentally determining the reaction rates at various substrate concentrations. This equilibrium constant Km is usually called constants or Michaelis constant. It is a measure of the affinity of an enzyme for its substrate. So in this graph, uh, so you can see this is the velocity, and this is the concentration of the substrate, and this is the progression. This is the product formation. This is the rate of reaction. So at particular time, so you can see here, it is rising, and after some time, we get a straight line. We get a straight line. So as the concentration of the substrate is increasing, the velocity of the reaction is also increasing. But when the substrate is saturated, it means when all the active site pockets of the enzymes are filled. So at that time we'll get a steady state kinetics. We get a steady state kinetics. So there is no increase in the velocity of the reaction. The final form in case of a single substrate is the equation, Michelson-Mentane equation, where V is equal to Vmax into substrate concentration divided by Km plus concentration of the substrate, where Kcat is turnover number, how many substrate molecules one enzyme molecule can convert per second, that is called as Kcat. Km is Michaelis constant, and an approximate measure of substrate affinity for an enzyme. During steady state, the maximum velocity Vmax occurs when all of the enzyme is in the enzyme substrate complex and is dependent on the breakdown of that complex K into Es. The microscopic meaning of Km and Kcat depends on the details of the mechanism. When V is equal to half Vm, Km is numerically equal to the substrate concentration. Or in other words, Km is equal to the concentration of the substrate, which gives half of the numerical maximum velocity. So when V is equal to half, then Km is the concentration of the substrate. So it is noteworthy that for any enzyme substrate system, Km has a characteristic value which is independent of the enzyme concentration. So Km value is used as a measure of enzyme's affinity for its substrate. The lower the value of the lower the value, the higher the enzyme's affinity for the substrate, and vice versa. Km value also provides an idea of the strength of binding of the substrate to the enzyme molecule. The lower the Km value, the more tightly the substrate is bound to the more tightly bound the substrate is to the enzyme for the reaction to be catalyzed, and vice versa. Km value indicates lowest concentration of the substrate the enzyme can recognize before reaction catalysis can occur. And this is the effect of substrate concentration. So this is, if I take the concentration of the substrate here, and what happens when we have enzyme and substrate, if I am going to increase the concentration of the substrate, the reaction progresses, the velocity increases, and at particular time this graph becomes a straight line. So it means when we are increasing the concentration of the substrate, the progression of an enzyme substrate catalyzed reaction progresses, but at one particular time it shows steady state kinetics, as there is no more increase in the activity or the reaction velocity of the enzyme substrate catalyzed reactions, because all the active sites of the enzyme are blocked with the substrates and they have to be converted. So no more free active sites of the enzymes are present. So it shows a steady state kinetics. And see here, if this is the maximum velocity or Vmax, and if I consider this as half V max, then if we pull this here, and this concentration is called as Km or Michaelis constant. So when reaction velocity is half Vmax, then Km is equal to the concentration of the substrate. So this is all about a brief introduction about enzyme kinetics, where we have discussed about equation, then we have also discussed what is Km, Kcat, and we have also discussed Vmax. So with this, thank you one and all.